Okay. So I'll just go over the answers, and then if you have any questions, let me know, but you can just double check yourself here. So the first part was with flipping a coin, and you had 2 coins, and so it gave you your observed data, but you flipped 200 times, or they flipped 200 times, so your expected for each one of these should be 100 and 100. So when you fill out the table, coin A should have a chi-square of 1.28, and coin B a chi-square of 0.8. So then when I asked you what is the hypothesis that has been tested, and that the hypothesis is that there's an equal chance of flipping heads or tails on a coin. And how we're going to tell if that hypothesis is correct or not is that if there's no significant statistical difference between the observed and the expected, which is the purpose of performing a chi-square test. So that's what you're doing here for number 2. The sentences about what you're doing, you're flipping your coin 200 times, they're recording how many are heads and tails, and then performing that chi-square analysis. And for number 3, we're flipping it 200 times. Alright, so that's that. So then when we flip this over here, we've already done 4 through 6 on the front side. It's filling in the data tables. But 7 asks you if you had to predict if one of the coins was a trick coin, which coin would you choose? It should be coin B. And the reason why is coin B had the furthest deviation from the expected. And so it was that one was probably most likely to be the rigged coin. So then 8, here's your chi-square symbol. 9 was filling in the chart on the front. So 10, you had to finish the sentence. So a larger chi-square value means the observed data is, and you should have very different from the expected data. So basically, when doing a chi-square analysis, the smaller the chi-square number, the better or more accurate your data is compared to the expected. All right, so small chi-square numbers are good. So then number 11 asks you, it starts to refer with the genetics problem in 2 heterozygous individuals. So this would be the Punnett Square if you're crossing 2 heterozygous individuals. And so you would expect in the genotypes a 1 to 2 to 1 ratio. So B asks you to, if you had 100, how many would be each genotype? So it would be 25, 15, 25. Okay? So then, performing the chi-square analysis, it tells you what your outcome from the experiment is, how many of each genotype you have. So it asks you to use a table similar to the other ones that we've used. So I made a table like this and put our 3 different genotypes and put the observed numbers of each one of those genotypes. Then we just figured out in the previous question the expected. So the expected would be 25, 15, 25. You figure it out, add these columns together, the O minus C squared divided by E for each of the 3 categories, and you get a chi-square number of 4.32. So then, letter D asks you about, in your opinion, if you look at the observed compared to the expected, do you think that your observed data is significantly different than the expected? And justify your answer. So you could put anything here and justify it, but the thing is that I want to point out is that really based upon looking at it, you can get a feeling, but until you do a statistical analysis test on it, you can't tell just by looking at it if it's statistically the same or different. So I have a little star here that this is a judgment call. Really, to truly answer that question, you need to do some analysis like the chi-square, okay, which is what we're doing. So then number 12 is about degrees. This is about degrees of freedom and so on. So number 12 is about the effect of the coin flip. So how many outcomes in the coin flip experiment there are 2, heads or tails. So therefore, how many degrees of freedom there are? There's 1. As the category is -1. And then with the genetics problem, there were 3 outcomes, 3 different genotypes, and so therefore 2 degrees of freedom. All right. So then they give you a critical values chart. This looks different than the critical values chart. It's the same data in it, and actually this one has more data in it than the chart I gave to you. The chart I gave to you is what is given on the AP test. But this gives you the same information. So 14A what's along the bottom of the table, that's your p-values or probability values. In the chart that I gave you in the previous thing, the p-values were along the side. And then B, what's along the side here, this is the degrees of freedom. In the other chart that I gave you, the degrees of freedom were along the top. But like I said, it gives you the same information. So let's see, I should use this a little bit. So what's the chi-square value needed to have a p-value of 0.5 in an experiment with 2 degrees of freedom? So we go, here's the p-value, 2 degrees of freedom is here, so you need a 1.39 there. All right, so then it's asking you to analyze the chi-square table here. So it says which p-values for A here, which p-values in the table indicate that the difference between the expected and observed data was likely due to chance. So chance means that there's not really something at play in that we can accept our hypothesis. And so that is any of these p-values from 0.95 all the way to 0.1. These are considered not significant. So that means that while your observed is different than your expected, it's not significantly different. And so therefore, in general, what that means is it can be due to chance and we can accept our null hypothesis. Whereas for B, asks you to, says which p-values in the table indicate that the difference between the expected and observed data was likely due to an environmental factor. That means something else that's going to change the data and then it's not just different from the expected data due to chance. And that is these p-values here from 0.05 all the way to 0.001. And so these guys are significant. So if you get, let's say you have, remember this is the column, in our other thing it was the row, the 0.05 is the p-value that we look to. And so if you had a degree of freedom of 1, and look at the 0.05 here, you need to have a chi-square number of less than 3.84, because if you get less chi-square value of less than 3.84, that means that that the percentage of time that we would see that same data it goes higher. All right, so any of this we can accept. If it's this or lower, we reject. And so then number 16 asks you if you have a p-value of 0.7, what does that mean in terms of the percent chance that the data sets are different only by chance? And so if you have if your chi-square number is down here where it would give you like a 0.7 p-value, what that means is you have 70% chance if you were to repeat the experiment, you could get the exact same data set. Okay. All right. So that's that. And so this would be acceptable. Alrighty, 17. As the chi-square values get larger, is the data more likely to be significant or not significant? So as the chi-square values get larger, if you look at the trends here in the table, as they get larger, they get less, more and more significant. All right, until you get to where these ones here, when you actually there's significant enough that you reject the hypothesis. 18, when an experiment has more degrees of freedom, is a larger chi-square needed for a significant outcome or a smaller one? And so as the degrees of freedom go up, the chi-square numbers go up as well. Okay, so that's 19, if we were talking about the coin flip experiment, which row of chi-square values would be used? And that would be the first row with 1 degree of freedom, because the coin flip experiment has heads and tails, -1, so you have 1 degree of freedom. 20 says find the p-values for each coin experiment, and if the chi-square value falls between 2 points on the chart, give a range of p-values. So for coin A, the answer, the p-value of that would be between 0.2 and 0.3. So let's talk about why that is for a second. So coin A had a chi-square value of 1.28 and 1 degree of freedom. So if we look at our chi-square chart, 1 degree of freedom, and we're looking at the 0.05 here. And so we're looking for a chi-square number of under 3.84. So it is under 3.84, and if we actually go to where 1.28 would actually be, a chi-square number of 1.28, that's between 1.07 and 1.64. So therefore, that's how we get between the p-value between 0.2 and 0.3. And so for the same reasoning then, coin B, the p-values would be between 0.01 and 0.001. All right, so then A, which coin produces a non-significant value that we can accept then, and that's coin A, and B, which produces a significant chi-square value, and that is coin B. So coin B is the one that we would reject. So look at here, it says therefore due to an environmental factor. So what could be an environmental factor skewing coin B's results and why they're so off from a 50-50 heads and tails? And so that's what C is asking you to propose a suggestion. And so they alluded to the fact that there's like a trick coin or something like that. So an example would be if the coin is weighted on one side, like heads versus tails, it would cause it to flip either heads or tails more often. And that would skew your data. So that's what it means by an environmental factor and why we don't have heads and tails unequal. So then 22, referring back to the mating question from question 11, does it indicate that an environmental factor was affecting the outcome? And justify your answer. So the chi-square value of problem 11 was 4.32. And there was 2 degrees of freedom. So if we go 4.3 and 2 degrees of freedom, go back to the chart here. 2 degrees of freedom, we're using the 0.05 column. So we're looking at, it has to be below 5.99. Ours was 4.3. So 4.3 is actually between 0.1 and 0.2. So what that means is our data for that experiment, we would see again between 10 and 20% of the time. So this is not significant, which means we can accept our hypothesis. All right, and so that's what means. This is not significant. All right. So then 23 is a genetics problem. Can you guys hear Mr. Claver all the way over there? So 23, you have to do this genetics problem. You're crossing a purple and a white flower, and you got, it told you got 124 purple and 82 white flowers. It asked you to do a chi-square test to test the null hypothesis that the purple flowered parent was heterozygous. Even if they didn't tell you that hypothesis, that should be your hypothesis, because if you cross a purple and a white plant and you get purple and white in your kids, that must mean that the parent had to be heterozygous. So this is what your cross would look like. So you would expect purple and white kids, but look at the ratio here. It's a 1-to-1 ratio. And so we have to look to see how far do we deviate from that 1-to-1 ratio. So I've made a makeshift little chart here. So here's my observed 124 and 82, 124 purple and 82 white. That gives me a total of 206 offspring. In my Punnett Square, I would expect a 50-50, a 1-to-1 ratio. So I divide 206 by 2 and put 103 in each category. Then I fill in the rest of the chart and get my chi-square number of 8.56. So then I use my chart and say, okay, my chi-square is 8.56. My degree of freedom is 1. So go over here, degree of freedom of 1, using that 0.05 category here. I want my chi-square number to be below 3.84, but my chi-square number is really high. It's between 0.01 and 0.001. It's 8.5 here, so it's between there. So therefore, my results are significantly different than the expected, and so therefore I would reject my hypothesis. All right. So what does that mean? When you reject the hypothesis, what that means, it necessarily doesn't always mean that the parent is not heterozygous. So the parent may be heterozygous, But there might be some other factor previously in here that they call an environmental factor that can affect your results. So let me give you an example of an environmental factor that could affect your results. So it could be that there's a disease that affects the white flowered plants and not the purple. And so therefore, you might have your results skewed with more purple versus white or so on. And so that would be an example of an environmental factor that could affect the results. And not just, so it doesn't necessarily always mean that your cross is wrong, all right, that there's some other factor in play. All right, and then number 24 was with a dihybrid cross. So we're looking at two traits at once, and they didn't tell you the cross. So it just says you performed a dihybrid cross of plants and get the following data. And it tells you that they got 206, well, I'll do down here, 206 purple and tall, 83 purple and short, 65 white and tall, and 30 white and short. And so looking at this here, in my experience with dihybrid crosses, this kind of looks like a 9:3 to 3:1 ratio. So my thought process was, okay, it kind of looks like a 9:3 to 3:1 ratio. What kind of cross gives me a 9:3 to 3:1 ratio? And that is a cross where the parents are both heterozygous for two traits. And so if we do that here, so our cross, I used R's and T's, big R, little r, big T, little t, cross with big R, little r, big T, little t. When you do that, I have the work here, the Punnett Square. When you do a Punnett Square between two heterozygous parents, you make your big Punnett Square here with your 16 boxes. You put your 4 possibilities for each parent. These guys are identical. You fill in your 16 boxes. What happens is 9 out of those 16 are going to show both dominant traits. 3 out of those 16 are going to show a dominant for one and a recessive for the other. And 3 are going to show the opposite, recessive for that one and dominant for the T gene. And then 1 is only going to show all recessive, and that's your 9:3 to 3:1 ratio. And so that's what I think. So then how do I fill in the expected here? So I have a total now of 384 kids. And when you cross these two heterozygous, there's 16 possibilities in their kids, or 16 different boxes. So what you could do is take 384 and divide by 16, and that gives you 24 in each group. And so it's a 9:3 to 3:1 ratio. So you could say, okay, there's 9 groups of 24, so 9 times 24, I would expect 216 purple and tall, which is both dominant. 3 groups of 24, so 24 times 3 gives me 72 here. Same thing here. And then all recessive is just 1 group of 24, so there's your 24. So then you get your difference and then square it divided by the expected, add them all together, you get a chi-square number of 4.325. And so this is your chi-square number. Your degree of freedom is 4 minus 1, so your degree of freedom is 3. So then you go back over to your table, so 4.3, degree of freedom of 3. So here's your degree of freedom of 3, and we're looking at the 0.05. So we want our chi-square value to be under 7.82. And ours was under that. So ours was 4.825, so that means we can accept it. And so our data is, we cannot reject our hypothesis. Any questions? Most people did pretty well on this. I looked at this on everybody, so most people did pretty good. All right, very good. Alrighty, so we are going to go back into our you guys, when you do me a favor, I just realized I lost you. Will you just look around your table? I was passing out paper, so I think maybe it might have stained me. Here's my one that's not lost. Is it in your skirt? I looked in my skirt. I should probably take it off and shake it out. No, because I was hoping that for looking. Oh, look it right, it's right here. Thank you for looking. All right. Last hour informed me I never finished the last. Did we finish all the 15 notes?
Well, I mean, I didn't, never talked about it.
Okay, do I need to just like spend a second for that?
Oh, this is what goes on. But we already did.
Were there some random chapter 15 I can't stick
Okay, so we're talking about genes that are found outside of the nucleus, and those are genes in the mitochondria as well as the chloroplast. They have their own genes. And remind me, did I draw an egg on the board and sperm and talk about that? All right, and how when the sperm fertilizes the egg, that the sperm is basically just a donor of the genes, and the sperm doesn't give any of the organelles. So for us, in all of our mitochondrial DNA, so here's the mitochondria. Mitochondria has its own DNA that's involved in making proteins that are involved in cellular respiration to make ATP. And so when you have a zygote, after the sperm fertilizes it, the baby now has 23 chromosomes from the mother and 23 chromosomes from the father, but then the baby also has DNA in the mitochondria that just came from the mom. So basically the fertilized egg cell, all of the baby's organelles were originated from the mother. All right, and so therefore any of the mitochondrial DNA that a baby has, and therefore even an adult as they grow, came from the mother. All right, and so that's what it means by the zygote. Cytoplasm comes from the egg and all the organelles as well. And so, so the same kind of thing goes with plants. A plant cell has two organelles that have extra DNA besides the nucleus. So plant cells have the mitochondria, so they have the mitochondria with its own DNA inside, and then they also have the chloroplast with its own DNA inside. And in addition to the nucleus that has its own DNA, okay? And so these are the extranuclear DNA. And so with the chloroplast, they studied this with plants that have splotches of yellow and green on them. And so basically, in the plant, the spots that have yellow, the plant cells, those plant cells in that spot, in their chloroplast, their DNA codes for making yellow protein, and so therefore it's yellow in that spot. And the spots that are green, in the plant cell, the chloroplast have the green gene to make the green protein. And so therefore it's kind of random what cells have where, so you get these splotches all over the place. All right, so that's what that means. And so the last thing is going back to us with our mitochondria here. We have diseases that affect our muscular and nervous systems that originate, they're caused by defects or mutations in our mitochondrial DNA. And so the mitochondrial DNA is codes for making proteins that have to do with cellular respiration. So therefore, if people have mutations in that DNA, they're not making the proper proteins and therefore may be deficient in some way in making ATP. There's a whole host of genetic diseases based upon mutations in mitochondrial DNA. So this DNA is different in addition to our 46 chromosomes in our nucleus, okay? And so, so those are mitochondrial genes, all right? And that's the end of chapter 15. All right, so now we're gonna, the guest teacher on Tuesday gave you the chapter 16 stuff, so maybe we get that out. And we continue on with this DNA stuff here, and look more specifically at DNA. Let me talk a minute about kind of what this chapter is about. So we know that DNA is the genetic material. We've talked about it with cell division and genes and giving traits to our children and so on. So this chapter is really in two parts. All right, the first part is how we figured out that DNA is the genetic material. We take it for granted that we know that DNA was the genetic material, but earlier on in this century, we didn't know that DNA was the genetic material. In fact, most people used to think that protein was the genetic material, all right, at one point. So it had to be proven, and it was a really big deal to prove that DNA is the genetic material. We've come such a long way since we figured out that DNA was the genetic material and all the things that we can do and manipulate with DNA now. So that's the first part in kind of the history and the story. The College Board, people who write the test, want you to know kind of the history of who the major players or scientists and what they did in the discovery of DNA. So that's the first part here, is just some history and some famous experiments. And then the second part is once we figured out that DNA was the genetic material, what's the structure of DNA, which we've kind of already talked about. We'll do a quick little review. And then how the structure led them to figure out how DNA replicates. Why is it important that DNA replicates?
So the offspring can get like the one copy and pass it on throughout generations.
Okay, so the offspring, you can pass on copies to your offspring. So that's like through meiosis, making egg and sperm cells. Yeah, and cell division to grow. Remember, we all started out being a zygote, one cell big with 46 chromosomes, but then that cell has to grow and divide, and you need to have two cells with 46 chromosomes. So we have to double the DNA. How do we do that? We replicate it. So we've already talked about DNA replication and the purpose for DNA replication, but the second part of this chapter is going to talk about what exactly happens in your cells to replicate that DNA. So how do we get an exact copy of that DNA? So that's the kind of the overview. So we'll look at some major people, Watson and Crick, I'm sure you've heard of before. They came up with the structure of DNA. They introduced the double helical model of DNA, or deoxyribonucleic acid. We are going to talk about how they figured out that DNA is the substance of inheritance. It's what's passed on from parent to offspring, and that the hereditary information is actually encoded in the DNA and is actually reproduced. And this DNA codes for and directs all of development and all of your traits, including, physical traits, anatomical traits, biochemical traits, makes enzymes and so on, all your different, even to behavioral traits. And this is Watson and Crick, and this is the actual picture of them with their double helical model that they came up with. All right. So 16.1 is, like I said, the story of the history of how we figured out that DNA is the genetic material. So this is a big, identification of the molecules of inheritance was a big deal, a big challenge. And so Morgan, remember the fly guy, Morgan's group showed that genes are located on chromosomes. So he was the first one to do that. The two components of chromosomes, DNA and protein, became candidates for the genetic material. So once Morgan did that with the flies and said, okay, this gene, he proved that, a certain gene is on the X chromosome or on a particular chromosome. So then once he did that, then we studied chromosomes and said chromosomes are made out of two things. They're made out of DNA and protein. So if genes are passed on to kids on these chromosomes, where are these genes at? Are they on the protein part of the chromosome or are they on the DNA part of the chromosome? And so they didn't know which one that is. And so now we know that DNA is wrapped around these histone proteins and that the coding or the genes are on DNA, but they didn't know that. So that's why DNA and protein were the two major candidates. So a key factor in determining which one was the genetic material was choosing the right organisms to do experiments on. And so we're going to look at some experiments done on bacteria and some on viruses. So the role of DNA and heredity was first discovered by studying bacteria and the viruses that infect bacteria. Viruses are, it says viruses that infect bacteria. Viruses are specific in who they what types of cells they infect. So not every virus will affect every living thing on the earth. And so there's a certain classes of viruses that infect only bacteria and wouldn't infect our cells. All right, so the beginning of the story starts with this man named Frederick Griffith. He worked with two strains of bacteria, a pathogenic S strain and a harmless R strain. What is pathogenic? Yeah, it's harmful. So it usually makes you sick or causes some type of illness. It's harmful to whatever bacterium is in, so if it's in us. And he actually looked at a strain of bacteria called Streptococcus pneumonia, which it's the bacteria that causes pneumonia in mammals. All right, so he was looking at different strains of them, and so one, the S strain, well, I'll tell you in a minute why they call it the S strain, and then the harmless, the R strain. So he's studying them, and he discovered something, and we'll write about it, and then I'll talk about it we'll look at a picture here. He killed remains of the pathogenic strain with living cells of the harmless strain. And he found that some of the living cells became pathogenic. And so, I'll show you a picture of this in just a second. He called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation of foreign DNA. What does assimilation of foreign DNA mean? Alan? What was that? That's right. So taking in DNA from another cell, foreign DNA, and incorporating or bringing it into its own DNA. All right, so you can think of incorporation as another word that can fit there. All right, so this picture kind of explains what those last couple bullets are talking about here. So let's look at this. This is living S cells and living R cells. So these are the two strains he was looking at of this particular type of bacteria. The living S cells have this kind of like little capsule here. He called them S cells because they're smooth, so the outside is smooth. And these guys do not have that capsule, and these guys he called them R cells, like rough. All right, so that's where the S and the R came from. And so if we look at when he studied them, he took these S cells and cultured them, put them in a syringe, and injected that culture into mice. And that's how we found out these guys were harmful. So the mouse died. All right, so this is your pathogenic. So these guys are pathogenic. And then he took the R cells, cultured them, injected them into the mice, and the mice is healthy. So these are non-pathogenic, or harmless. Here, because the mouse stays alive. So that's what he did. Then he started experimenting a little bit. He took the living S cells and he applied extreme heat to them. He called them heat killed. So he killed them. They weren't alive anymore. And so by applying extreme heat, so he killed them, injected them into the mouse, And these mice stayed healthy. So therefore, this also was harmless. So when he applied and heat killed them, what was once pathogenic became non-pathogenic by heat killing. And then what was interesting is that he then combined a couple of these. So he took the heat-killed S cells, which by themselves are harmless. He took the living R cells, which by themselves are harmless, mixed them together. So he mixed two harmless things that didn't kill the mice, mixed them together, injected it into the mouse, and the mouse dies. So there's a conundrum. He's wondering what in the world is going on here. So he opens up the mouse and he cultures some of the bacteria in the mice, and he puts it on a petri dish and grows the bacteria and looks to see what's in the mouse that's making it kill. All of a sudden, what's in the mouse are the living S cells that he never put into the bacteria. All right, so his question is, where in the world do the living S cells come from? Because I didn't put those in there. And upon further study, what he found out is that when he heat killed these S cells, so that they're harmless, in the solution, in the heat killing process, it kind of can denature the cell a little bit, and some of the DNA can leak out. So therefore, you have the S cell DNA floating around, all right, not contained in those heat-killed cells. And what he found out is that bacteria, in all bacteria this is the case, bacteria, if you take a bacteria cell in a solution and throw in some DNA, that bacteria takes up foreign DNA. Bacteria do that, all right? They take up foreign DNA surrounding them. Our cells do not. If you took human cells and put it in some solution and threw in DNA by itself, our cells do not take that up. In order to get DNA in our cells, we have to physically put it in there, all right? And so our cells do not do that, but bacteria cells do. And so what did that bacteria, these R strains, do? They took in this S cell DNA. So the living, I can write this out. So the living R cells took up the S cell DNA. And if it has the S cell DNA, then guess what? It can make the same proteins as the S did as well. So guess what? It actually transformed into an S cell because it has the S cell DNA. Does that make sense? Okay. So therefore, the living R cells took up S cell DNA and transformed into S cells. And S cells, so now that it has these guys have the S cell DNA, it can make proteins that make it pathogenic, and it kills the mice, and that's why we see these guys here. So these guys here, even though I didn't put them in here, these used to be the harmless R cells, all right, before they took up the DNA. And that's called transformation, and that's why the definition, when we say transformation, the genotype and phenotype has changed. So this living R cell, its genotype has changed because it took in foreign DNA, so now it has genes it didn't have before. And then because it has genes it doesn't have before, it can make proteins it didn't have before, and therefore its phenotype has changed, and it became a living S cell. But what Griffith didn't do in this experiment is prove that it was the DNA. So he said that there's transformation occurring, but this doesn't prove, he didn't know for sure what was taken up by the R cells. And we knew it was the genetic material, but he didn't know exactly what it was. And so some other groups of people, Avery, McCarty, and McLeod, did some further study and announced that the transforming substance was actually DNA. So that's what changed the R cells into the deadly S cells. Their conclusion was based upon experimental evidence that only DNA worked in transforming the harmless bacteria into pathogenic bacteria. So they did a little bit of further research. So that's the first evidence really that DNA is the genetic material. But a lot of people were still really skeptical. There wasn't a lot known about DNA. Still, most people in the field still thought protein was the genetic material. All right, so in comes some other people working with viruses. So more evidence for DNA as the genetic material came from studies of a virus that infects bacteria. These viruses that infect bacteria are called bacteriophages, or short, often they're called phages. And they're widely used in genetic research. We're gonna, in later chapters, you'll hear these bacteriophages come back into play. This is what they look like. So, so this is an actual electron micrograph picture of 3 different bacteriophages in blue here, infecting a bacteria cell. So the bacteriophage has this head portion. This is called the tail portion, then it has these little tail fibers. And what they do when they infect a bacteria cell is they inject its genetic material into the bacteria cell. So this is what this is here. This is the DNA being injected in here. Viruses, what they do, viruses by themselves cannot reproduce. Viruses, if you have a whole bunch of viruses hanging out, they can't make any more viruses. They can't reproduce on their own. They need a cell, and they call it the host cell. And so this is what they're doing with the bacteria. So how they use the bacteria is they inject their genetic material, and now the bacteria has viral DNA inside of it, and they basically make the bacteria, they use the viral DNA and make new viruses. So the bacteria ends up making new viruses, and that's how viruses reproduce. We'll talk more about that a little bit later, but that's why they're injecting the DNA, because they can't reproduce themselves. And so, so when we look at a bacteriophage, I want to talk about, this is the cell, and here is the phage, the head, the tail, and the tail fibers here. This part right here, there's 2 major components. This kind of outside shell, sometimes it's called the protein coat because it's made out of protein. So a bacteriophage is made out of protein, and then inside of that head region, it has its genetic material, which the bacteriophage, the genetic material is DNA. So a bacteriophage is mainly made out of 2 things, DNA or protein. And so what we wanted to figure out is, what is the bacteriophage injecting into the bacteria? Is it injecting the protein, or is it injecting the DNA into the bacteria? And so these were good organisms to use because they're made out of just basically 2 things, protein and DNA. This little guy here, when I always look at the bacteriophage, always reminds me of the shape of it. Reminds me of, you remember in Toy Story, and there's the evil kid next door, and he takes that, what makes that toy with the head, and then the spider legs. The nerd in me, when I saw that movie, was like, oh, it's a bacteriophage. When I see that little toy, looks like a bacteriophage, and that's how it kind of looks like to me, anyway. And so, looking at what they did to study the bacteriophage, Hershey and Chase, they did an experiment with a particular bacteriophage called the T2 bacteriophage, and this infects E. coli bacteria. So to determine the source of genetic material in the phage, they designed an experiment showing that only one of the 2 components of T2, the phage, DNA or protein, enters an E. coli cell during infection. So this is the T2 and this is the E. coli. Which one? Is it the protein or the DNA that actually injects? That's what they designed an experiment to do. And their conclusion, as we might expect, is that it was the DNA that's injected. All right? And therefore the bacteriophage's genetic material is DNA. So here we have, this is their experiment. So this is the Hershey and Chase experiment. It's kind of busy here. There's 2 parts to the experiment. This part here, so we're looking at it in rows, and then this part here. So I'm just going to draw a line separating them. All right, so these guys are different. So notice that it says batch 1 and batch 2. And then batch 1, it says sulfur, and it says S35, and then phosphorus is P32. So remember that the purpose of their experiment is they're looking at the bacteriophage and saying, okay, is it DNA that's going in or the protein going in? So how are we gonna tell that? So let's look at what's different between DNA and protein. One of the differences is their atomic components, so what atoms they're made out of. So notice here, we have phosphorus and we have sulfur. Phosphorus is only found in one of those. It's in either DNA or protein. Do you guys remember? It is found in DNA. If you remember, just real quickly here, DNA has a backbone and it has the bases that come out. And so the backbone here, if you remember, is the sugar. We go sugar, phosphate, and then the base, A's, T's, C's, and G's that come out. And so the backbone is the sugar phosphate backbone. And so phosphate groups have phosphorus in it. So DNA has phosphorus. So I'm going to put in here, this is in DNA. And then sulfur, we can deduce, is only found in protein. Sulfur is in our groups of some proteins, all right? So this is in protein. And then if we look here beyond this where it says P32 and the 35S on there, what those are, they're isotopes of those atoms, so they're radioactive. So that's going to be important. So this sulfur here, this is radioactive. That's what this is supposed to be getting at. And same thing with the phosphorus. And the reason why they make them radioactive is so that they can see where they end up. Bless you. So in their, you know what, we're gonna run out of time here. So tomorrow, I'm gonna talk to you about what they actually did. All right, so we'll keep you on the edge of your seats.